It's getting nicer, right? People start not showing up, and so you have more help.
Oh, wow.
It's always the case. It's always the case that, for the most part, good students show up, and students who don't do well for some reason don't show up. Maybe that's anyway. So we have two more sections. Two more sections to talk about. One is cellular adhesion. Okay, let's see. Remember, this set of exam is about cell structure and structure everything that's within the cell. Also, it's about how a cell interacts with the other cells as well as an extracellular matrix. Then, so that's cell adhesion is this part, and then we'll leave the good stuff for last, which is talking about vesicular transport, where we'll take everything that we learned for this set of exam and look at how material is moved around within the cell, okay? Which includes cytoskeletons, which includes talking about cell adhesion and so on and so forth. We will later on talk in more details about cell adhesion. So right now it's just a kind of a hand wave, a quick look at cell adhesion before we go into vesicular transport. So don't be surprised that there won't be too much information for now. This slide right here, you need to know most of this slide, okay? Especially the first part right here. Okay? So if you want to have it memorized, fantastic. I'll go into a little bit more detail for the next 10 or so slides, and then later on we'll go into even more details about these things. All right. So cell adhesion. Cell adhesion refers to the fact that cells have to physically interact with other cells, as well as physically interacting with the extracellular matrix. Okay, so these are the various types of cell adhesions: anchoring junctions, occluding junctions, better known as tight junctions. If you ever took a course that talks about tight junctions, that's in a nutshell occluding junctions in animals. Channel forming junctions, we talked a little bit about it in terms of the gap junctions, and then we talk about the signal relaying junctions in terms of the synaptic signaling. Right? So we already talked about these two. We'll focus on these. And what makes a junction, or what makes a junction, it requires what is referred to as a transmembrane adhesion protein. So all of these junctions require a transmembrane adhesion protein. Okay? All right. And so again, we're going to focus on these. Let's see, for the anchoring junctions, we're going to look at cell-cell junctions versus cell matrix junctions. Okay, there are two types of cell-to-cell junctions and two types of cell-to-extracellular matrix junctions. Now, you have two types because each junction has a different function. So it's not just a junction, but what interacts with what, right? A cell interacting with another cell, but what does that interaction, what is its function? Okay? So again, there are two types of cell-to-cell junction and two types of cell-to-extracellular matrix junction, and they all have specific functions. We'll talk about adherence junction. We'll talk about, it's called actin-linked cell matrix adhesion right here. We'll call that focal adhesion or focal contact. So that type of junction is called focal adhesion or focal contact. And then we'll talk about a desmosome and a hemidesmosome. Okay? So we'll go with that first, and then we'll talk about occluding junctions, where in vertebrates, it's the only occluding junction is tight junction. We're not going to talk about invertebrates at all. Okay? And then these others, we'll talk about gap junction and plasmodesmata, and also the chemical synapse. We'll talk a little bit about how in the immune system, how that works as well. Okay? So let's get started. The first example is a hemidesmosome. A hemidesmosome is a very specific anchoring junction. There are 4 types of anchoring junction, and hemidesmosome is one of it. And hemidesmosome is formed between an epithelial cell with a basal lamina. That's it. That's the only time that you can find an hemidesmosome is between an epithelial cell and a basal lamina. Remember when we talk about epithelia, that an epithelium is always going to anchor onto a what? A basal lamina, right? So an epithelium anchors on, or epithelial cells within an epithelium that anchors onto the basal lamina. That's done through a hemidesmosome. Okay, and to dissect that, there are 3 parts to all anchoring junctions. Now, when we talk about gap junctions and synaptic junction, those are slightly different. But for anchoring junctions and tight junctions, all of them have 3 parts to them. Right. The very first part is something called a transmembrane adhesion protein, which in this case, for hemidesmosome, the specific transmembrane adhesion protein is integrin, or integrin, however you pronounce it. There are many ways to pronounce these terms. Okay, so there's a transmembrane adhesion protein in the form of integrin, and then on the extracellular side, hemidesmosome interacts with the extracellular matrix. In this case, it happens to be the basal lamina, not just basal lamina, but a particular protein in the basal lamina called laminin, not lamin. Remember lamin? Lamin is a subunit for the nuclear scaffold. So this is different from laminin, which is the extracellular protein that is the major component of the basal lamina, where integrin binds. So integrin binds on the non-cytosolic side to laminin, and on the cytosolic side, integrin actually gets anchored onto, in terms of hemidesmosome, gets anchored onto the intermediate filament. But integrin, the cytosolic domain of integrin, does not have a binding domain for intermediate filament, so it has to anchor onto the intermediate filaments via proteins we call intracellular anchoring proteins. Plectin and dystonin are examples of intracellular anchoring proteins. So again, the 3 components of an anchoring junction are a transmembrane protein, in this case it's integrin, a protein where proteins that are bound by the non-cytosolic domain of the transmembrane adhesion protein. In this case, it's laminin. And then on the intracellular or cytosolic side, the cytosolic domain of the transmembrane adhesion protein anchors onto the cytoskeleton, but through or via the intracellular anchoring proteins. So those are the 3 components. Okay. Adhesion protein, extracellular protein, and the cytoskeleton. Okay. So that's a desmosome. Very specifically for a desmosome, integrin has to be integrin is the transmembrane adhesion protein. Laminin is the extracellular component that integrin anchors onto, and intermediate filaments is the cytoskeleton that integrin has to anchor onto, but again, not directly, but through an intracellular anchoring protein. Okay. So a few things, and the same idea will follow through all the other anchoring junctions. So I don't have to repeat this, but the idea is this: how does a cell know that if it's a hemidesmosome that the outside must be laminin and the inside must be intermediate filament, right? It's because the cytosolic domain of a transmembrane protein, any transmembrane protein, not just adhesion protein, but any transmembrane protein, the cytosolic domain and the non-cytosolic domain actually talk to each other, right? They actually talk to each other. Okay, so we say that there is linked recognition. Both domains are linked to each other. So whatever happens to one side, right? Whatever happens, let's say, to the non-cytosolic side, will tell the cytosolic domain what to do. So let's say this integrin was formed, and then it is bound to the laminin first. By binding to the laminin, right, that causes conformational change, which then tells the cytosolic side to change its conformation so that it can anchor onto intermediate filaments instead of anchoring onto another cytoskeleton. Does that make sense? And then by anchoring onto the intermediate filaments, then the cytosolic domain will tell the extracellular domain or the non-cytosolic domain that, okay, everything is good. Now this is a hemidesmosome and not anything else. And by being a hemidesmosome, then that tells the cell that this performs a certain function. Does that make sense? So the linked recognition is really important, and where somewhere here we have that term somewhere. Right, there you go. So linked binding or linked recognition, right? So again, internal and external binding are linked. Okay, meaning that because of the change in conformation, you see how powerful that is. That's why I introduced the idea way like from the very first lecture practically, that conformational change is very powerful. Okay, so again, the non-cytosolic side and the cytosolic domain talk to each other by conformational change. Okay, one. Another important thing about all of this is that the hemidesmosome allows the cell to interact, to hang on to the extracellular matrix, but it has to be, but the cytosolic domain must be anchored onto a cytoskeleton. Why is that? It's because you see this integrin here, it's embedded into the plasma membrane, right? The plasma membrane is fluid. So what happened to the integrin? It might float around. If it floats around, then that interaction right here can be broken. So the integrin or integrins multiple have to be fixed in place. How are they fixed in place? How are they fixed in place? How do you think they're fixed in place? By anchorage onto the cytoskeleton, right? So by anchoring onto the cytoskeleton, the integrin is fixed in place within the plasma membrane. So anchorage to the cytoskeleton is very important. Now, we're going to find out that some anchoring junctions anchor onto intermediate filaments, and other junctions anchor onto the microfilaments. Nothing anchors onto the microtubule. So why do you think so what would be the function of the anchor if it were to anchor onto the intermediate filament versus the microfilaments? Roughly, what do you think? What's the difference between anchoring onto one versus the other?
One is dynamic, one isn't.
I'm sorry?
One is dynamic and one is not.
Right, exactly. So hemidesmosome binding to intermediate filaments, you would think stable, stability, strength, right? So that's exactly what it is that a hemidesmosome provides strength, not to the cell. It has nothing to do with the cell. It has to do with the whole tissue. Okay? So if it were my skin, right, and I pinch it off like this, it provides strength so that when I pinch off my skin, that the epithelium doesn't fly off, right? So it provides mechanical strength or tensile strength to the tissue. The tissue, not the cell. It has to do with the tissue. So hemidesmosomes provide mechanical or tensile strength. Please, when you put it, you can put it way over there. Don't walk in front of the screen, please. Provide mechanical or tensile strength to the tissue, right? So that's what hemidesmosome does. Okay? All right. When I do this, okay, my skin can fly off, so I have to have mechanical strength, but there has to be flexibility in the tissue. So some anchoring junctions can provide flexibility to the tissue as well. Okay? And that is what focal adhesion or focal contacts do. Is that focal adhesion or focal contact is also a cell to extracellular matrix interaction. Okay, so both hemidesmosome and focal adhesion form cell to extracellular matrix adhesion. It's just that focal adhesion or focal contact, instead of providing strength, mechanical strength, it provides flexibility to the tissue. Okay? And because it's cell to extracellular interaction, it's still integrin. So integrin provides cell to extracellular matrix interaction. Okay? A few more things about this slide is the fact that, again, on the extracellular side, the matrix side, since its focal adhesion performs a different task, therefore it binds to a different components of the extracellular matrix. In this case, it's collagen instead of laminin. So if a cell were not an epithelial cell, and if a cell were, let's say, a fibroblast or any other cell that is found in loose connective tissue, and there's a lot of collagen fiber in loose connective tissue, right? That's how a cell hangs on to the collagen fiber, or that's how a cell settles down in the loose connective tissue by forming focal adhesions or focal contacts. Okay, also again through integrin. But binding on the extracellular matrix side to the collagen. On the cytosolic side, integrin is actually anchored onto actin or filament or microfilament instead of intermediate filaments. Why? Because by providing flexibility, then the focal adhesion must be more dynamic, right? Depends on a cytoskeleton that is more dynamic. And microfilament is dynamic. You see that? So there's a connection there. Okay, so if you try to remember which goes with which cytoskeleton goes with which adhesion junction, anchoring junction, then you think about stability or mechanical strength versus flexibility or dynamic. Okay, that's how you do that. More about this is you notice that integrin doesn't act as a single subunit. Okay, integrin only acts as a dimer. So you have to have two integrin molecules. Okay, and they're they form hetero dimers. Okay, so two different subunits of integrin. Okay, so that's a minor. A more major component of this slide is the fact that for integrin, whether it's hemidesmosome or focal adhesion, you notice that integrin interacts with some other proteins, right? So we call this heterophilic binding, meaning that a protein binds to something that is not itself. Later on when we talk about cadherin, actually cadherin likes to bind to itself, so it's homophilic adhesion or homophilic binding, right? So in this case, this is heterophilic binding because the transmembrane adhesion protein binds to a protein on the extracellular matrix side that is not to itself. Does that make sense? Again, I already talked about internal and external. So internal meaning cytosolic side, and external meaning the non-cytosolic side, and the two sides are linked. So if one is activated, the other is inactivated. Okay, and through what? Through conformational change. So desmosome. Desmosome is, again, using my skin as an example. So desmosome links a cell to another cell, not to the extracellular matrix. So now we're talking about cell-to-cell adhesion, and it's desmosome. And because it's cell-to-cell, the transmembrane adhesion protein is no longer integrin, but is cadherin. Cadherin, that's how you pronounce it. That's actually exactly how you should pronounce it, not cadherin or anything else. There's only one pronunciation for cadherin. Okay? So cadherin is that. So cadherin would allow a cell to interact with another cell. And cadherin undergoes homophilic binding, meaning that a cadherin can only bind to another cadherin that is found on the surface of the other cell, right? So cadherin over here binds to the cadherin over here. Cadherin would not bind to integrin or anything else over there, right? So cadherin performs homophilic binding. You see the difference between heterophilic and homophilic? So then, therefore, the extracellular matrix components that is bound by cadherin is another cadherin, right? And then what about the cytosolic components? Cadherin still must bind to or anchor onto a cytoskeleton for desmosome. It just happens to be intermediate filaments. So therefore, you should think that desmosomes then provide mechanical or tensile strength to the tissue again, never the cell, right? The cell is unaffected by what type of anchoring junctions you have, right? It's to the tissue. Versus adherence junction. Adherence junction is also a cell-to-cell anchoring junction. So therefore, cadherin is also used. So therefore, the extracellular components that cadherin binds to is another cadherin, because cadherin only form homophilic binding, right? However, cadherin is anchored onto microfilaments on the cytosolic side, so therefore adherence junctions provides flexibility to your tissue. So you see you have two cell-to-cell anchoring junctions. One provides flexibility, one provides mechanical strength. You have two cell-to-extracellular matrix anchoring junctions. One also provides flexibility, and one also provides mechanical strength to the tissue. Right? So they're kind of parallel. Okay? And there are actually multiple forms of adherent junctions that we won't talk about. We talked about desmosomes before, right? Remember the skin layer, the quantified skin layer, the spike cells, the spiky cells, the spike cells. The spikes comes from the fact that we have a lot of desmosomes. Okay. Let's see. And all these, there's a whole family of cadherin and a whole family of integrin also. So the specific members of the cadherin and or integrin families that a cell express, it's cell specific, right? So the expression of cadherin and integrin is cell specific. Why do you think it's cell specific? What does that mean by cell specific? So what if specific cells expresses specific member of cadherin protein or integrin? What does that do? What would be the relevance of that? Anybody? And the answer is not a trick question. This is not a trick question. Sometimes it is, sometimes it isn't. Why do you think that different cell type expresses a different member of the cadherin protein? Isn't it?
So only like cells that can
Yes.
Either like cells or cells that are meant to adhere to each other will adhere. Simple as that, right? It's as simple as that. Always ask yourself, why are certain mechanisms general and why are certain other mechanisms specific? Right? Always go back to that idea of DSCAD, the DSCAM. There are over 38,000 versions of the DSCAM protein. Right? Why do you need so many? Because you only want cells two cells to talk to each other when you want them to talk to each other. And if you don't want two cells to talk to each other, then you block, you prevent, you don't have the means for them to talk. So perfect answer. Tight junctions. So we're going away from anchoring junction. We're talking about tight junctions. Tight junctions has the same three components. It has an extracellular component, a transmembrane protein, a transmembrane adhesion protein, and then it anchors onto the cytoskeleton. the anchoring and transmembrane adhesion proteins that happen to be both claudin and occludin. Okay, even though there's a mix of claudin and occludin in tight junctions, but they also form homophilic binding. So claudins from one cell will interact with the claudin from the other cell, and occludin from one cell would interact with occludin from the other cell. And I can't remember, I always switch back and forth. I think that tight junctions are stable, so therefore I think that they're anchored onto intermediate filaments. But don't quote me on that, but I'm pretty sure that that's the case. But in any case, there is a cytoskeletal component to that. Okay? What's the function of tight junction? Function of tight junction is this, is to prevent anything from leaking through the epithelium. So tight junctions are really only found in an epithelium. It's found at the tip. We call this the apical end or apical side of the cell, the top side, and this is a basolateral side. So tight junctions are found at the very top, and think of it as a zipper, right? So these are tight junctions, and nothing can pass, not too many things. Now, water and very small molecules or ions might be able to pass through, but nothing, not too many other things can leak through. So tight junctions prevent leakage across the epithelium, right? Let's say this is your stomach. You don't want junk in your stomach to leak through the epithelium and get into your peritoneal cavity. Then, I mean, when that happens, that can be fatal, right? You don't want all that acid, all that waste to get into your peritoneal cavity. So tight junction does that. How about blood-brain barrier? You don't want things to cross the blood-brain barrier. How do you prevent that? Tight junctions. Okay, so that's important. What happens if you don't have tight junctions? There you go. You have things leaking through. Now, leakage across the epithelium or in between the epithelial cells, it doesn't prevent anything from getting into the epithelial cells, right? So it's not preventing anything to get in. Anything, how do materials get into the cell? By either diffusion or active or passive transport, right? So we already talked about that, okay? Okay, here's a special type and a fairly unique, integrins or focal contacts are fairly unique, okay? In that integrin can sometimes formed between two cells. So before we talked about integrin in the Henle desmosomes in the case of a focal contact, where integrin now is used for cell to extracellular matrix interaction, but integrin actually can bind to can help two cells interact through a process called rolling adhesion. Let's see, we'll talk about this a little bit more later, but I'll introduce it now. Later meaning the next set of exam, or the last set of exam when we talk about wound healing. But basically, to set up the scenario is that you might have an infection or you might have an injury, and so during for an infection you want to fight the infection. For an injury you want to carry out wound healing. Now, wound healing or getting rid of infection requires blood cells, right? Cells in the blood to move to that area to either again carry out wound healing or getting rid of infection. Then these cells then must have a way to get into the area, specific area. Well, blood actually flows really fast, so it's very, very difficult to have the blood cells flowing all out so fast, and then how do they get out of the bloodstream? Does that make sense? So almost like the equivalent is almost like driving down the highway and you want to make a right turn, and then what do? You go down, right? You don't go 70 miles an hour, some of you drive 100 miles an hour, and then you can't just turn, you have to do what, you have to slow down, and that's what rolling adhesion is slowing down. So blood cells are slowed down. How are they slowed down? Again, by this rolling adhesion process. The rolling process, okay, so the epithelial cells contains two proteins. One is called P-selectin or selectin, okay, and another one is called ICAM, which is not on here. Right, ICAM or CAM, okay, and CAM, C-A-M, is an adhesion protein. A stands for adhesion, is in the same family as, guess what? Antibodies, immunoglobulins. So immunoglobulins, most of the time when you hear about immunoglobulins, you think about antibodies, but not all immunoglobulins are antibodies. Some immunoglobulins are actually adhesion protein, right? So you have those two proteins. So you have the CAM or CAM, and then you have selectin. What happened is that in the rolling part, selectin is a protein that binds to carbohydrate residues on the surface of cells. Guess where we've heard about that before? Glycosylation, lots of carbohydrates on the surface of cells, right? So selectin then would bind to carbohydrates residue on the surface of the white blood cell. Now, selectin binds to carbohydrates but doesn't bind very, very tightly. So what happened is that a cell zoom by and brushes against the selectin, and that causes a temporary binding and then let go, and so that causes the cell to roll. That's why the cell rolls, right? It rolls along a bed of selectin. Think about that, and think about that image. To a point where on the surface of the epithelial cell is CAM, right? The NCAM or ICAM, the CAM protein. On the surface of the white blood cell, there are integrins. The integrin can also bind to the CAM proteins. And so in this case, that's the adhesion part, is integrin forms a very tight binding to the CAM protein. So that's the rolling adhesion. Now that the cell is stopped in its tracks, so rolling adhesion, then the cell now can undergo a process called extravasation, meaning to leave the bloodstream. Okay? And we'll talk about extravasation, and then we talk about the mechanics of it, which is diapedesis later on. But at this point, all you need to know is, so integrin is also involved in the rolling adhesion process. Did that make sense to anybody? Okay, yes ma'am.
So the integrin that's in the white blood cell
Mhm.
is what attaches to
On the surface of the white blood cell. Right. So then when the white blood cells are in the tissue, actually, they have to crawl around, right? So they also have to form focal contact. So the same integrin that help adhere the white blood cells to the epithelium of the white blood vessel will also be used to adhere to the collagen fibers in the loose tissue. Okay. So we'll get to this again with even more detail later on. Okay, you notice how I introduce material most of the time, a little bit at a time, and then so that I don't give you too much information right off the bat. Don't worry about this, except that it's ICAM or CAM, CAM, NCAM. They are important in adhesion. Large family. Okay? So that's adhesion. Like I said, we already talked about gap junctions. We already talked about synaptic junctions. We'll talk about them in more details later on. But for now, that's a quick look at adhesion. Any questions about adhesion? You probably learned a lot about it already. So this is a big one. This is a big one where we take a lot of what we already learned for this set of exam and for this class to explain what is called intracellular trafficking. So I always start out by telling a story about how when I was about to finish graduate school, to look for fellowship, that's basically an internship. And I was accepted into a lab up in Michigan or Minnesota where and this is in the early 90s, right? They were trying to understand how materials diffuse across the cell from one part of a cell to the next. That idea is absolutely ridiculous now, but at that time, people thought that proteins and materials just kind of diffuse from one area to the other. Now we know that we actually talked about calcium already, even ions are very specifically delivered to certain places. Most things do not diffuse within a cell, right? There is they move in a very orderly fashion. That's why it's called trafficking, right? Just like the traffic in the city, okay? And therefore, in order to direct traffic, you need roads. So the roads is the equivalent of cytoskeletons in this case. So both microtubules and microfilaments. Okay, so those are the analogy. So, and it's all intracellular. So we're talking about just about anything, including ions, but mainly protein complexes and organelles, like vesicles. So in terms of intracellular trafficking, like I said, it can move protein complexes around. So even if ions are moved around, but the ions have to be bound by a protein, which then would be moved around. But also, vesicles can be moved around. So a type of intracellular trafficking is something called vesicular transport. Okay? And then in moving vesicles around, which means that you may move mainly proteins around, then the vesicles are membrane bound. So we also say that this is lipid trafficking. So all of these different names depending on what molecules you're studying, right? If you're studying proteins, then it's just merely intracellular trafficking. If you're studying proteins that are moved around through vesicles, then now you call it vesicular transport. And then if you're studying the movement of vesicles, but you're paying attention or you focus on the lipids, now you're talking about lipid trafficking, right? It's the same process, but different names because depending on what your focus is, okay? So you need to know all of this. Luckily, I don't know if it's lucky or not, but I'm going to talk about each one of these in much greater detail, okay? But at the end of the day, why does a cell need intracellular trafficking? And these are some of the ideas right here, okay? A more philosophical idea or overarching idea is the fact that life that we think of in not just Earth but in the universe it requires water, right? Requires water. So we live in an aqueous environment, meaning that we live in a hydrophilic environment, but yet hydrophobic materials can still move around. So that's the idea, is that how do you move materials around that don't really like water? Or something like carbohydrates molecules, carbohydrates where they like water too much. How do you move them around without dragging all the water and dehydrate everything on its path, right? So how do you do that? So again, the more philosophical idea is again, how do you move basically things that are hydrophobic in an aqueous environment? And I don't know that I don't usually define terms because I assume that you know them, but when I say aqueous, that is the equivalent of water, right? Aqueous, water, same thing. So now we're coming back to talking about the endomembrane system, okay? And we're focusing on transport of proteins, okay? Transport of proteins. Transport of proteins that are either membrane bound or to be secreted. Okay, mainly, either membrane bound or to be secreted. They go through the endomembrane system, which will start with the nucleus and then we'll go on, we'll tick all these boxes all the way up to maybe peroxisome, and then vacuoles is more plant. So we're probably going to cross that out, and then we'll go to the plasma membrane. Okay. So here's the deal. Here's the deal. Let me see. There's a better picture. Yeah. Okay. So let me do this. Let me start with this slide, then come back, and then go forward. And I want to remind you that how the slides are set up is a kind of a compromise between Dr. Landerholm and me. But then when I lecture, I want to do it my way. So the slides are set up as a compromise, but when I lecture, it's so that it makes sense to me, and hopefully if it makes sense to me, when I tell you, it makes sense to you. That's why sometimes I skip slides. So let's start with the nucleus. Nucleus is where DNA is formed. And for DNA, now again, we're just talking about the nuclear genome. DNA will then, there are genes that encode for mRNA. Then mRNAs have to be modified or processed properly and then get taken out into the cytosol. And then the cytosol is where all translational event occurs for the nuclear genes, right? And then translation occurs, and then you have the polypeptide. Polypeptides or proteins that are to be membrane bound or secreted will have to be pulled into the ER, right? So that's back here. Now we're back here. Okay, they're pulled into the ER. We'll talk about very specifically how they're pulled in a minute. They're pulled into the ER, make their way from the rough ER to the smooth ER. And then what happened is that these proteins, whether again they're soluble or membrane bound, the ER membrane pinches off into something called a vesicle. And then that vesicle is transported, therefore vesicular transport, transported to the cis phase of the Golgi. And then get dumped in there. And then these proteins get gathered up, and then another vesicle gets pinches off, go to the next cisterna, right? So there are multiple cisternae, and then these proteins move from one cisterna to the next, all the way to the transphase of the Golgi. And then from the transphase, the vesicle forms again. The vesicle is either delivered to the plasma membrane, and that part right there is a part of the exocytotic pathway. Okay, so this part of the vesicular transport is also referred to as the exocytotic pathway. Those vesicles can actually move to the lysosome as well. Because why? Because there are proteins, lysosomal proteins, that must be delivered there. Okay. Some of these vesicles actually come back and bind to the Golgi again. Because in each of these cisternae, including the ER, there are proteins that must function there, right? So therefore, they have to be processed, go all the way to the transcisterna of the Golgi, and then they are properly processed, and then they are sorted out, and then delivered back to wherever they're supposed to. Okay? And then the peroxisome is actually cross-site peroxisome in this case as well. So vesicular transport, really, peroxisome is different. Proteins get delivered to the peroxisome actually differently. Okay? So the vesicles, as it says here, the vesicle acts as a bridge, as a moving bridge. So it's not a permanent, physical, stable bridge, but it's a moving bridge. Okay? Yes, sir?
I was going to say, you said the protein processing occurs at the transphase of the Golgi.
Processing of the protein occurs all the way up to the transphase of the Golgi. Right. Okay? Some other lysosome is also involved, and vesicular transport also involved with autophagy. Okay, so the cell, if let's say the mitochondrion in this case has been around for too long, for a few months, for a year or so, then the cell wants to recycle it. It's breaking down, so we want to recycle it. So the lysosome basically eats up the bits and pieces of the mitochondria. Okay, and then that is when peroxisome is involved, because remember the lysosome only breaks down macromolecules down to oligomers, and then the peroxisome actually undergo beta oxidation to break down all these oligomers into the actual monomers, right? So that's that, and then we kind of sort of looked at phagocytosis roughly already. Okay, so phagocytosis is also a part of the vesicular transport. Do I need to identify free ribosome versus bound ribosome? Let's do that. Okay. So translation occurs on free ribosome first. Always is initiated on free ribosome. You already saw that, right? The small ribosome is subunit. Just a small ribosome is subunit has to recognize and bind to an mRNA that whose the 5.3 prime end must be linked, right? Right. So it has to be free first. And for proteins that are to be again inserted into the membrane or to be secreted or to be delivered to, let's say, the lysosome, then the polypeptide must be pulled into the ER membrane. Sorry, I'm looking ahead. Okay, did we take those slides? It's okay. So in order free ribosomes then free ribosomes have to dock onto the cytosolic side of the ER membrane, right? Before they're pulled in. So when the ribosomes interact with the cytosolic leaflet or layer of the ER membrane, these are now considered to be bound ribosomes. Bound. They're not inserted into the ER membrane. They're just bound. And therefore, translation continues to occur in the cytosol, even though the ribosomes are bound to the ER membrane. Right? You see that, right? So translation is never in the loop of the ER membrane. So basically, that's the difference between free and bound ribosomes. So bound ribosomes used to be free ribosomes until they're told to become bound ribosomes. Okay? And what's the signal that indicates whether or not they should be bound ribosomes? Topogenic sequences. Right? So the topogenic sequences on the polypeptide is recognized by some protein, some membrane-bound protein in the ER membrane that then can be pulled in. So then the topogenic sequence, I'm sorry, not topogenic sequence. There's something called a signaling sequence. But anyway, there's some kind of a signal, right? But the point is that on the polypeptide, not on the ribosome, right? A ribosome is a ribosome is a ribosome. But different polypeptides would contain different signaling molecules, signaling sequences that would tell the ER that would indicate that these free ribosomes now must be bound. Okay?
And then because of like the chaperone protein?
Not chaperone proteins, the signal, the actual sequence on the polypeptide.
Okay.
It's actually called the signaling sequence.
Okay.
I forgot that we took out a bunch of slides that describe that part, which is actually, I always like to teach that, but no more. Okay.
Is that the polypeptide that is in the ribosome, or the polypeptide?
Possible constituents.
Ask that again. I think I know what your question is, but I'm going to get you to be clear on the question, and then you can answer your own question. So go ahead.
So the signaling sequence within the ribosome, the polypeptide that's in the subunits, the creatine.
Right. Where else would the signaling signal sequence be if it's not on the polypeptide? Right. That makes
Not
sense.
what is created. I was thinking maybe what is included here. Do you know what
No, it is the signal sequence is the amino acid sequence.
Okay.
If I say that is a signal sequence on the polypeptide, the only thing that's on the polypeptide is the amino acid sequence. So just the next couple of slides, just give examples of what type of proteins that are to be pulled into the ER versus the translation is completed in the cytosol, right? So bound ribosome means to be pulled into the ER. Notice receptors, transporters, they're all transmembrane proteins. Even the adhesion, transmembrane adhesion proteins that we talk about. And then obviously secreted proteins, and then enzymes that inside other organelles like lysosome. But even the Golgi, the ER, right? Versus free ribosome, meaning that these polypeptides will not be pulled into the ER, and anything that is in the cytosol, all the monomers for cytoskeletons, motor proteins. Later on, we'll talk about second messengers, everything that has to do with glycolysis, right? But not the Krebs cycle, because protein enzymes that carry out reactions for the Krebs cycle are actually encoded by mitochondrial DNA, right? So these are some examples. You know this. Okay, so let's see. Okay, so in the ER, why do proteins have to go through the ER and the Golgi first? I kind of talked about it before. Some of this is a review, but it's good to review. Lipid biosynthesis. Biosynthesis is how should I say that, is the use of lipids. For instance, lipid biosynthesis, protein biosynthesis, meaning that not just the translation of the protein, but processing, right? So if I say somewhere in here it talks about protein biosynthesis, not meaning translation. Translation has to occur in the cytosol, but protein biosynthesis, meaning the modification of protein, right, occurs in the ER as well as in the Golgi. Okay? And certainly lipid biosynthesis, meaning in terms of lipids, meaning that lipids are both synthesized in the ER, but also are used to be attached to proteins to form lipoproteins. So all of the above, all of those activities are under the term lipid biosynthesis, and that occurs in the ER. So therefore, that's why polypeptides have to be pulled into the ER. Okay. Also, like I said, membrane-bound proteins, and any proteins that have to have lipids as well as protein. That's just redundant. Number 3 is just redundant. So the ER is really important. Okay. All right. So now we're getting down into the nitty-gritty of how things work, how they are integrated into the membrane, how they move around, the cytosolic submembrane protein meshwork, and the varying lipid, what we call lipid signature and protein signature. Okay. So the idea is that anything that is on the cytosolic side, whether it's a domain of a protein, whether it's the particular phospholipids within that side, right, must be fairly constant, right? And so you will never have this protein right here switching side. So if a protein that must have its amino acid terminus in the cytosolic side, then it's always going to be put there. That domain is never in the non-cytosolic side, and vice versa. Okay? So we say that once cytosolic, always cytosolic. Once non-cytosolic, always non-cytosolic. Right? Proteins don't switch side. Okay? So therefore, if the carbohydrate residues are added to the protein in the non-cytosolic side, then you will always have carbohydrate residues interacting with the non-cytosolic environment and never the cytosolic environment. Okay? So that's what it says. Also then, in terms of the cytosolic submembrane meshwork, that it's a cytosolic submembrane meshwork, so therefore you would never ever find the cytosolic submembrane protein meshwork interacting with the non-cytosolic environment. Very important to note that. Okay, so Okay, so as vesicles move around, that's exactly what happened. Is that the non-cytosolic environment of the ER will also equal to the non-cytosolic environment in the lumen of the vesicle, and then the cytosolic environment of the ER membrane will also will also transfer into the cytosolic environment of the vesicle. So therefore, the ER membrane right here, on this side there should be the cytosolic submembrane protein meshwork, right? The same cytosolic cytosolic submembrane protein meshwork is also on the cytosolic side of the vesicle, which is then also on the cytosolic side of the Golgi apparatus. Does that make sense? So again, once cytosolic, always cytosolic. Once non-cytosolic, always non-cytosolic. Okay. So in terms of vesicular transport, always remember that. Okay, that cytosolic, non-cytosolic always are separated. Okay. There are four major events that occur during vesicular transport. The first is budding. Okay. The second is transport. The third is targeting, and the fourth is fusion. And all of these requires all of the components that we already talked about. We talked about the type and ratio of phospholipids that are found in the membrane. Right? Have we identified lipid signature and protein signature yet? Oh, okay. So let's identify them before we go on. So there are a number of different types of phospholipids, right? And so each membrane contains a very particular phospholipids, as well as a very particular ratio among the different phospholipids that are in there. Okay, does that make sense? So let's say just say the plasma membrane only contains phosphatidylinositol and phosphatidylethanolamin. Okay, but then it will contain 80% phosphatidylinositol and 20% phosphatidylethanolamin. So if you as a scientist, or if the cell sees that there is 80% phosphatidylinositol and 20% phosphatidylethanolamin, guess what? That's an indication that this is a plasma membrane. So therefore, this is referred to as a lipid signature, right? Just like your signature. It identifies the type as well as the ratio of the various lipid molecules in that membrane. So far, so good? That's a lipid signature. A protein signature is the fact that each membrane also contains different types of proteins at different ratio. And not only that, but again, once cytosolic, always cytosolic. Once non-cytosolic, always non-cytosolic. So therefore, the proteins are not only in the membrane, but have to be in a certain direction. So that's called the protein signature. Okay? So lipid signature and protein signature. Okay? There's more to the story. So lipid signature is not just the lipid signature of each membrane, but each leaflet has its own lipid signature. So the non-cytosolic leaflet of the plasma membrane has a different lipid signature than the cytosolic leaflet of the plasma membrane. So again, so that we can determine that once cytosolic, always cytosolic, once non-cytosolic, always non-cytosolic. So you know which leaflet is the cytosolic leaflet of the plasma membrane and which leaflet is the non-cytosolic one. Okay? Protein the same, right? So there might be different proteins of different ratio, but again, the protein points in a certain way. The protein signature also also is a signal of the leaflet of the particular. So doesn't just indicate which cell membrane it is, but also indicates which leaflet that you're looking at. Very particular, right? So lipid signature and protein signature are very important for vesicular transport. Okay? In addition to that, then, the recognition of a cytosolic submembrane protein meshwork is also important in far so good? Yes? No? Okay, so let's go on. I'm gonna go. So you need to know this again, as always. So I have one slide that tells you everything that you need to know, and then I'll break them down for later slides. So you have budding, you have transport, you have targeting, and you have fusion of the membrane. Okay. So budding first. Let's say that this is the ER. ER, and that's a lumen, and this is a cytosol. Okay, and these are the proteins, and this green one also. These are all proteins that must be transported outward. Okay, so budding. What happens to budding? Budding is when certain proteins recognize that this is not. First of all, recognizes that this is the ER membrane. Okay. And how does it recognize that this is the ER membrane? How do these recognition proteins that help in budding, recognize that this is the ER membrane? I'll give you a hint. There are three things that it recognizes. The same three things I just talked about. Lipid signature, protein signature, and cytosolic submembrane protein meshwork. Right? Those are the three things that are recognized by so that budding can occur. So that these proteins, we call them coat proteins. These coat proteins won't go to the Golgi apparatus first. Okay? So they'll go to here, and the same coat proteins that recognize the ER membrane also induce budding. So almost like I always think about a balloon, that you have a puff-up balloon, and then you suck on the skin, and then you get a little smaller bit, right? I always think of that. So that's called budding, okay? And then eventually that part is going to be severed. So the membrane is going to be we call that a neck. The neck of the vesicle will be severed, okay? And then you get this vesicle that is delivered to the next. So that part is targeting. Right? So budding and then targeting. How does the cell know that vesicle needs to go to a target? Let's say the cystostern of the Golgi. How does it know that? Because of lipid signature, protein signature, and cytosolic submembrane meshwork as well. And there are proteins that regulate that. Right?
Okay.
And then the membrane of the vesicle will fuse into the membrane of the target compartment. Okay, due to mainly protein signature and definitely lipid signature. Okay, by this time the submembrane protein, cytosolic submembrane protein meshwork don't really do much. So again, it's mainly the protein signature and the lipid signature that allows for the fusion. Okay. Even more detail now. Detail. So budding first. There are these proteins called coat proteins. Clathrin is a type of coat protein. There are two others. There are three. There's COP1 and COP2. COP. COP is an acronym for coat protein. So COP1 and COP2. So there are three different types of coat proteins. All coat proteins do the same thing. They identify the organelle where budding must occur, and then they catalyze or they induce the budding process. But the reason why there are three different coat proteins is because there are different organelles. Each coat protein recognizes a different membrane. How does it recognize the different membranes? Again, the same thing: lipid signature, protein signature, and cytosolic submembrane meshwork. By saying clathrin, I know that this organelle this membrane can only be either the membrane of the transcistern of the Golgi or the plasma membrane. Can't be any other membrane, right? So again, the particular coat protein that is used will identify very particular membrane. Okay, so whether it's clathrin or anything else, we'll just say that it's a coat protein. What the coat protein does is that again it induces the budding. That neck right there, the next slide will show that there's a protein called dynamin that actually sever. Dynamin is a long protein that wraps around the vesicular neck, and just squeezes it until the vesicle just pops out. Okay. Now this vesicle is referred to as a coated vesicle because it's coated with coat proteins. If the coats remain there, what do you think happens? Coat is pretty thick and it covers the entire vesicle. What happens if the coat remains there? Then the lipid signature, protein signature, and the cytosolic submembrane protein meshwork won't be recognized. So therefore, the coat proteins have to be removed through a process called uncoating. Now you have a naked transport vesicle that contains your transport protein, or sometimes called a cargo protein. Okay? Don't worry about the adaptive protein. That's something too specific. Okay? So far so good? So the budding process is again due to the coat protein. I won't go into more detail than this, by the way, so this is the level of details I need you to know. So what you need to know is that the coat protein recognized the various signatures that are on the particular membrane and carry out budding. Once budding occurs, then the uncoating has to occur so that the naked transport vesicle is exposed. But then it's important to have a naked transport vesicle is because, again, all the signatures can now be recognized, right? So that you have targeting. So targeting, so still coat protein. So roughly, again, don't try to memorize which one does what. But as you can see here, COP1 will only transport vesicles from the ER to the Golgi. You see that? I mean COP2, only that. It doesn't work anywhere else. And then COP1 is used to transport vesicles throughout the Golgis, either in that direction or that direction. Okay? And then both COP1 and clathrin are involved in budding from the transcisterna of the Golgi. And then only clathrin is involved in budding, invagination of budding from the plasma membrane going the other way. So when we talk about endocytosis, only clathrin is involved. Okay? Again, don't try to memorize all of this, just to give you an idea of why all the signatures are important. So that COP1 will never ever bind here and carry out budding because the signatures aren't there. Okay? So that's dynamin is what the thing that causes breaking. So targeting. This is just a reminder that the next step is targeting. Again, we have so many redundant slides. Targeting. So transport. I'm sorry, budding and then transport. So transport. How are these vesicles transported? Right? How are they transported? They are transported mainly along the microtubules. Sometimes, though, they can be transported along microfilaments if transport occur in the cell cortex. Right? So when you talk about, let's say, exocytosis or endocytosis, then vesicular transport in that case, in that area, can be along the microfilaments. But for the most part, when vesicles are transported from the ER through all of the Golgi, right, then it's done along the microtubule. And when it's done along the microtubule, then if you're moving outward from the nucleus toward the plasma membrane, then since microtubules radiate out and polymerization occurs toward the plus end, therefore outward meaning along toward the plus end of the microtubule, therefore kinesin is used as a motor protein. And going the other way, if there are proteins that must travel from the Golgi or the plasma membrane toward the ER, then if it's still along the microtubule, then dynein will be used, right, to carry vesicles toward the minus end. Sometimes you can say, toward the plus end means toward the plasma membrane, and toward the minus end meaning toward the nucleus or inside. I hesitate in saying that is because You can say that for microtubules, but when you talk about microfilaments, it doesn't work that way, right? So I prefer to talk about plus n toward the plus n versus toward the minus n. Okay, so what do molecular motors or motor proteins recognize for this? They recognize the proteins that are found within the cytosolic submembrane protein network. They don't really recognize either the lipid signature or the protein signature. They actually, motor proteins recognize the cytosolic submembrane protein network. Okay, and I think that should be the next slide. Yeah. So this is an example of a diamine that recognizes all of these things that are found in the cytosolic submembrane protein network. Okay, and binds to it, and that's how it knows when to move. Let's talk about targeting, and then this is a good point where I talk about it now, and then I'll repeat it later on because it's a little bit more complex. So targeting. How does a cell know? How does a cell know that this vesicle needs to target to its target compartment, right? Even though we know that, well, lipid signature and protein signature, but what are those lipid signatures? What are those protein signatures? Okay. On the membrane of all vesicles and all organelle membrane, there are two proteins, right? One is called Rab, and one is called snare. Okay, and both of them are important for targeting a vesicle to its target compartment. So let's talk about snare first. There's a V snare. V stands for vesicle, and there's a T snare. T stands for target. So the V snare of a particular vesicle will recognize the T snare of its target. Okay. But the snare proteins aren't that specific. So the main function of the snare really is eventually, once the vesicle had reached its target, that the snare proteins will ensure that the vesicles stay bound to the target membrane. Does that make sense? So the V and T snare, still there's some specificity there, but not a lot, right? But the main function of the snare proteins are to allow the vesicle to hang on tightly to its target protein. It is actually the Rab proteins that allows for the specific targeting. So there's a Rab GTP, and there's that Rab, which is a G protein. Rab protein that will target a certain Rab effector. I would call that a Rab protein itself, right? So there's a Rab protein on the vesicle and the Rab protein on the membrane of the target membrane. This is what we refer to as the protein signature, right? So a particular Rab GTP on the vesicle indicates the protein signature of that vesicle versus the Rab effector protein on the target membrane signifies the protein signature of that membrane, right? And so that's how targeting occurs, is mainly through protein signatures, mainly through the protein signature of the Rab and snare. And the last thing is the fact that there are many, many different Rabs. Each one is associated with a different vesicle. It's obvious, right? Snare, there are a few different ones, but like I said, snare, B snare, and T snare aren't too specific. B snare is specific for T snare, but then there are only a few members of a B snare and few members of a T snare. Okay, but definitely there are a bunch of different Rabs to allow for very specific targeting of vesicle to target. Okay, so have a wonderful weekend, and I will.